So, hello. This is the actual title of this is, what is the title of this one, Paul? Are we in a antibiotic-free era? So, this is a critical issue. I mean, so first off, let me introduce myself. My name is Andrew Robertson. I'm actually a substitute moderator here. I've been called in the last minute, but I'm really glad that Paul asked me because this is something that I work in industry right now, and this is something that our company has actually been very much engaged with over the past few years. So what I'm going to do is I'm going to say a few words from the private sector perspective and go to our three panelists and ask them to give a few minutes, 10 to 15 minutes of from their perspectives in the lab. And then, but honestly, I think this is a conversation. I mean, this is a roundtable, and so for questions that you all have, please feel free to actually raise your hand, and we'll incorporate those definitely into the discussion. So, for myself, I work in a relatively young field in industry. It's called regulatory science, and what we do is we're really concerned with like the laws, the policies, the science, the standards, the strategies that are involved with drug development, drug approval. Some of the questions that we ask: are there ways that we can develop a drug faster? We can make sure that it's safe. Can we get it to patients and provide greater access to some of these treatments that are really potentially life-saving? Like I said, I work in the private pharmaceutical sector. If this panel was yesterday, honestly, I would say that I would tell you that I work for a company called Merck, but I resigned from there about 12 hours ago. But Merck is a great company. It's in 2015. Relevant to this panel, in 2015, they paid almost $10 billion for a company called Cubist, which was almost entirely focused on anti-infectives and antibiotics. Likewise, they got late-stage development of a drug called daptomycin, which is one of the more recent drugs for MRSA. They also have a number of vaccines, such as Pneumovax, to prevent pneumococcal infection. And then on top of it, they have a couple of monoclonal antibodies in development for targeting diseases like C. diff. So that was as of Friday. Now, as of Monday, I'm going to be joining a company called Sanofi, and they're doing also as regulatory science, but they are going to be looking at, they look at diseases like Zika, dengue, and also pneumococcal infection as well. So this panel, we're here to discuss antibiotic resistance, and I think it's a critical issue. And if you just look at the numbers, it really kind of underscores how important it is. CDC numbers in the U.S., they pen about 23,000 deaths annually right now, with 2 million treated. They estimate about $20 billion in direct healthcare costs, with another $35 billion in lost productivity, and that's just in the U.S. Globally, there's about 700,000 deaths annually by estimates. Now, if we don't act, which is the other important thing, then very quickly manageable diseases like gonorrhea are going to become untreatable. We'll see a loss of global GDP of about 100 million per year, and then about 10 million deaths annually, or 1 every 3 seconds if we stay on the course that we are. Likewise, this has been made a healthcare priority by many major countries worldwide, most Western regulators, including the EU, the UK, the World Health Organization, Germany, and of course the US. So, what we do in regulatory science is we say, okay, well, this is all well and good, but what can we actually do about it? We've had a lot of discussions here, and I think, honestly, the path forward has been broken down into 3 main categories, and I'd love to hear the panelists' perspective on this. But from our perspective, we see the following 3 areas. First is better use of the tools that we have. That means, employment of vaccines to actually start preventing instead of treating. Proper stewardship, not only in human health, but also in animal health as well and in agriculture. And then even more broadly is this issue of communication with patients and healthcare providers to mitigate this feeling of fear of if you go in with a viral infection, not to automatically ask for an antibiotic as well. So that's the first one. The second is to actually accelerate the development of new tools that we have. This is actually very hard, but what we're talking about is addressing the regulatory paradigm. Are there ways to actually create clinical studies so that it's not this huge high burden or this huge high barrier towards market access? But likewise, if we were to actually get it approved, are there ways that we can get approvals but still have responsible use of these new drugs? And for example, in the EU, they have things called adaptive approvals. And in the US has been floated this concept of special medical use, where you would get a drug approved, but only in certain conditions and in certain instances. The other issue, too, is incentives. And really, and this is hard to underscore enough, but the current business model just really doesn't work. It's not a drug that you would take through the lifetime of a patient. It's something you would take for hopefully, only a few weeks, maybe a month or so. It's not as, infectious disease doctors are actually some of the lowest paid among medical practitioners in the US. So we need to actually start looking at this from an incentives point of view as well to get more industry engagement. We have seen some success. There's this one program called the Qualified Infectious Disease Program in the US where they've actually added additional exclusivity for approved drugs. So that's the second thing. And the third is, and I think this is the more interesting part, but it's really to investigate new approaches and new therapeutic paradigms. And this includes things like probiotics, phages, immune modulators, looking at getting bacteria to actually target harmful pathogens, targeting resistance genes, basically thinking outside of the box. We're really using almost 1950s technology here in this fight against bacterial infection. So can we actually start thinking of new ways to do this? And then I'm going to say something. I was at an Institute of Medicine conference a couple of months ago, and one of the experts there is a gentleman named Stuart Levy. He actually gave, I thought, a really interesting introductory comment. He said, okay, first off, this is a very difficult challenge. And first off, we're outnumbered physiologically. Depending on how you look at it, the number of bacteria in your body outnumber us either between 3 to 1 to 10 to 1. The estimates vary, but bottom line, our cells versus bacterial cells, we're outnumbered. Second is we had a late start. Prokaryotes have got about 3 billion year head start on us. So a lot of these genes that they've actually developed to help avoid many of these antibiotics, we're catching up. And then last is we're depending on strategies which, like I said, are almost 100 years old. With Alexander Fleming, the penicillin is basically the foundation for a lot of the antibiotics that we're using. That was 1928. So we're about 90 years down the line without any huge developments, with maybe the exception of monoclonals. And again, I'd like to defer to the panelists on this. So the questions he said is, look, is this a losing battle? Do we need to have a new strategy? And really, should we focus past antibiotics? And I like that question because it brings us full circle to this panel, which is, are we in a post-antibiotic era? So with that, I'll just go down the line and then let Peter go.
So I'll take kind of half a minute just to introduce who I am. So I did my PhD here as a Gates Scholar in 2001 in bacterial pathogenesis, but looking from a slightly different angle. So how bacteria adapts to colonize a new host. In that particular case was Yersinia pseudotuberculosis, which is a mammalian pathogen, but towards Yersinia pestis, who actually interacts with an insect to then infect a mammalian cell or mammalian host. From that, I kind of went completely off tangent to protein engineering, synthetic biology, and now I'm kind of coming back to work with phages, and sort of I can see myself migrating back into antimicrobials. And to some extent, kind of what I want to bring to the table are sort of some of the new things that are coming out of synthetic biology that could be applied to this question. In terms of the questions you posed, I think sort of it might be consensus here. Are we living in a post-antibiotic era? No, not yet. But we will, unless we change the framework, unless we change the way we tackle the problem. We mentioned that we have 3 to 1 bacteria, but they're not all the same. It actually also changes with age. So there's a much more complex scenario there, and one that if we tackle the problem that way, we might be able to find different answers. Also in terms of kind of evolutionary, from an evolutionary perspective, what we're trying to do is give a poison to bacteria. That's usually easy to escape. But what if you give something that will have, sort of to escape, they have to become sort of less pathogenic? So potentially the idea of targeting the resistance gene, and we might come back to that example later. And I'll be tempted to say that's it, and kind of keep the discussion.
Okay. I was actually asked to produce some slides, so I did. So I apologise that... Okay, so my name's Mark Holmes. I'm a lecturer here at the vet school in Cambridge. I'm actually a reader in Microbial, Genomics and Veterinary Science. I trained as a vet. And I just want to give a little bit of perspective of antibiotic resistance in agriculture. So there are many different aspects to being in an antibiotic-free era, but one of the most important things is antibiotics are not finished. They are absolutely vital, absolutely essential for dealing with a lot of diseases, both in humans and animals, and they actually work pretty well most of the time. And the reason we're getting a bit scared is every now and then we get an accumulation of antibiotic resistances in a particular pathogen, and suddenly you've got somebody in hospital, had a hip replacement, had tumour therapy, and we've actually only got one or possibly two antibiotics that can possibly treat them. And we also know that of those antibiotics out there in the sort of microbiome in general, other resistance genes could be accumulated that would make this disease untreatable. And I think there are some very key facts. And I understand pretty well everything I understand in terms of disease from the point of view of evolution. And that is, whenever you use an antibiotic, you are providing a natural selective pressure, or selective pressure as in natural selection, and you are going to pick those bugs that are resistant. And one of the things I always have to remind non-veterinary audiences is it's you who've caused the problem, not the animals. Generally, almost all antibiotic resistance of significance in human hospitals is a direct result of antibiotic use in humans. Okay? So in terms of scale of problem, getting doctors to prescribe, getting human doctors to prescribe properly is probably, in the order of sort of 10 times more important than getting the vets to do it. Having said that, it's really important to appreciate that antibiotic resistance arising from livestock does enter the human sort of pathogen or bacterial population, and that profligate or inappropriate use of antibiotics is an issue. And the one thing that that everybody knows about and is clearly a stupid thing to do is using antibiotics to increase the efficiency of animal, livestock production for food. So if you give low levels of antibiotics to cows or pigs, they grow faster. Okay, and that's great for the farmers. It's bad for antibiotic resistance. And my work has been looking at antimicrobial resistance in bugs, and I was just describing to the panel a few years ago, a PhD student, so this could be happen to you, just happened to stumble across, literally, a new form of MRSA that we found in cow's milk. It gained massive amounts of publicity. It gained me a lot of research money, so I am eternally grateful both to the student and to the bug. And you'll be aware of the recent discovery of MCR1, Colistin resistance, which was one of the last resort antibiotics for human use. Why is that of relevance to us? What happened when Colistin was developed? Colistin was a polymyxin that, when it was developed, it was found to have neurotoxic effects and renal toxicity effects in people. So suddenly the human doctors, the farmer, they suddenly said, well, this isn't actually turning out to be so good. It's knocking out kidneys and it's turning people's brains to mush. I tell you what, why don't you vets have it? And as it turned out, it was quite useful in veterinary medicine. It's very effective against respiratory disease problems in pig farms, and pigs don't live long enough to get dementia. They don't live long enough to get their kidneys knocked out. So they were happy to leave it to us. In the meantime, through excessive use of antibiotics in human beings, they ran out of antibiotics. And they suddenly remembered they had this colistin that was a bit problematic, but it seems that all the bugs we're worried about are still susceptible to it. So suddenly they said, we'll better use this as a last resort antibiotic. And in the meantime, of course, we've been using it badly, profligately for growth promotion, particularly in China, and that's why we ended up with resistance. If you think about how antibiotics are used in farm animals, they're often used in groups. And if you get resistant bugs and they end up on the meat that we eat, and we don't prepare that meat properly, we don't wash our hands when we're dealing with the raw meat, we don't cook the meat properly, we can end up infecting ourselves. And with MRSA, that happens. There are livestock-associated MRSAs. They end up in meat and they end up in people. And we've shown that in every country that we've looked at, including Britain. But what's worse is that all the antibiotic that doesn't get degraded in the animals or in the farm ends up in slurry or manure, gets spread on the ground and for crops, production of crops. And we provide yet another way of getting it into the human population. And not only that, we're exposing the maximum possible microbiome, in other words, almost the world microbiome, to the antibiotics that we overuse. And clearly, that is not a good thing. So what do I think are some of the topical issues? Better antibiotic stewardship is a no-brainer. So don't go around chucking antibiotics everywhere. But it's important to appreciate that we don't know enough to even use the antibiotics we have got intelligently. We often go straight to use of antibiotics, and including broad spectrum or reserved antibiotics, because we fear that bug may be resistant to a narrower spectrum bug. And the reason we do that is it takes us at least 24 hours to get resistance or susceptibility data. So getting accurate diagnosis and rapid identification of resistance is actually a key thing, because probably 90% of the time we could have used an antibiotic that was less crucial. We don't actually understand what happens to the antibiotics in bodies half the time, particularly in veterinary medicine. So when you have a group of animals, be it chickens or pigs, it is not economic to go and give a pill to each of those animals or even to inject each of those animals. Most of the time, the antibiotics is given in the feed and in the water. If you're a sick pig, you need the antibiotic, and you're in a pen and lots of the other pigs actually haven't got that disease yet, or they're at different stages in disease, they'll get the lion's share or the pig's share of that feed. And so what you do is you're actually you're ending up underdosing the very animals that need the antibiotics. And more than that, we don't even understand the pharmacokinetics or pharmacodynamics of these antibiotics when they're given in food, because most of the regulatory work is done on laboratory animals. Even when it's done on the animals that are the target animals, they are done using injected antibiotic, not antibiotics given in food. So a lot of information, basic information, not exciting, high-tech, monoclonal antibody, genomic science, but just understanding how antibiotics are distributed around the body is important. And in fact, one of the questions that nobody has ever been able to answer to my satisfaction is, why do you give an antibiotic for 5 days? Why do you give it for 7 days? Why do you give it for 2 weeks? Why do you give it a month? Sometimes it's obvious, because there's a bloody great pus exuding out of a wound. It's obvious your animal or your patient is still infected. But for many diseases, we say, you have to take this course. And what does your doctor tell you? He says you have to finish the course. Well, why? What's the evidence that says you generate less antibiotic resistance by continuing with the course? Because on one hand, I can see that if you give a very short pulse or you give lethal doses of antibiotics, you're possibly going to increase the rate of mutation, you're going to increase selective pressure. But actually, for most disease where we use antibiotics, your immune system is the thing that actually cures you. And what you do with the antibiotic is you just push the balance away from the pathogenicity of the bug to give the host's immune system a chance. So we need to do a lot more basic research, I think. And one of the things we don't know about is what happens to all these genes that are responsible for antibiotic resistance when they're out in the world, in your gut, on a field, in an animal that you're about to eat. When do they jump between species? When do they jump within species? What makes them jump? How often do they jump? How likely are they to survive afterwards? What are the costs associated with all these antibiotic resistance genes? Not in the laboratory, not with a single bug in a simple experiment, but in the complexity that is the whole environment. And we mustn't forget that we can't do any of this research that's going to help inform the decision-making we need to make to preserve our antibiotics without considering the global context. So I can argue to my heart's content in Britain and persuade the politicians to stop doing things or ban things that I think are unsafe. But people will always complain, says it doesn't matter what you do in Britain. In China, in India, in Nepal, in wherever it happens to be in the world, these are unregulated drugs. Their use is unregulated. And quite apart from the fact, I think we should do everything we can when we can afford to do it in the West as an example of good practice. But we should be thinking about how we can help places that do not have the same infrastructure and that have different financial or cultural or political influences make their decision. So that's my introduction. I'll hand over to Paul. I'll help him by getting his slides ready. There, you'll know he is.
Thank you. And so for those who don't know me, my PhD is essentially in how bacteria swim, and I look at the protein dynamics that underline that process that builds a nanostructure that's been studied for around 100 years called the bacterial flagellum. So if you remember to high school or whenever the last time you took biology, it's the thing that rotates, leading to swimming or swarming motion among bacteria. And so just kind of Andrew touched upon this earlier, this idea that bacteria have had a long time to develop this resistance. And I think something we have to remember is a lot of this resistance happened in the absence of anthropogenically produced antibiotics. So the resistance was already in the environment for a variety of reasons, either because a lot of the antibiotics we're using in the clinic were derived from soil bacteria, particularly actinobacteria, and so other bacteria already had time to develop resistance to this, and also the hypothesis that early on antibiotics may have been used as signaling molecules to other bacteria to kind of indicate what environment they were in. And so you have this very long evolutionary time span that bacteria have been interacting with antibiotics, and that then challenges us when we use it in the clinic or when we use it in agriculture, as everybody else has said, to think about how we rationally and intelligently use these. And so from a molecular basis, my background as an undergrad was to look for new antibiotics using a technique called metagenomics, which is where you don't culture bacteria like we do traditionally, but you look at the DNA that's in an environmental sample. And a lot of that was to look for antibiotics to overcome resistance that's being built, and primarily by essentially these 8 steps. And I mean, it's not really important that you guys know what each one is, but it's more just bacteria have a huge arsenal to use against antibiotics. They can degrade them, they can change the target, they can overproduce the target protein or whatever. They can pump them out of the cell, they can sequester them in a location in the cell where the antibiotic target is not located. And so that's where the molecular world comes in, and where, as Mark said, more basic research is needed. So if you look at this beautiful protein structure, this is a 3-part efflux pump. So the whole purpose of this protein in bacteria is to move molecules from inside the cell out of the cell. And it looks very pretty. It's made up of 3 different proteins that span 2 membranes. So if you remember back to biology, cells are protected by membrane. Well, some bacteria have 2 of those, so you have to span both cellular spaces. And in this one's case, it's called the ACR-AB Toll-C pump, which was actually solved here in Cambridge, as an interesting side fact. You can see all of the different chemicals that this is responsible for pumping out of the cell. And in fact, in general, this pump basically removes any small hydrophobic molecule from inside the cell. So any small hydrophobic molecule that you will introduce to bacteria through as an antibiotic or as a small drug molecule target, this pump and others like it have the potential to pump that out. And we've seen as soon as you start overexpressing, or as soon as you introduce antibiotics or any type of small hydrophobic to the environment, bacteria will overexpress these types of pumps, and then resistance is a short way away. And so that's kind of where I'm coming from in the molecular world, is just an emphasis that more basic research is needed to understand. You can't necessarily block this, right? Because bacteria will find ways around that. This is critical to their daily lives. And I think we have to understand that. If you design an antibiotic that is going to put great stress on the bacterium to change, then because their time scale is so much shorter than ours, they will change. What you can do through combination therapy and very intelligently designing molecular targets is slow this down. And if you can slow it down, then, as Mark said, your immune system can begin to clear that bacterium from you and end infection. So that's something that I think we should keep in mind. And just briefly, the molecular kind of approach is certainly not the only one out there, and we do need to keep in mind that I would agree that we're not in a post-antibiotic era, but I think we have to think beyond that as well. And so going back to this, we need more basic research to address the question of bacteria. A lot of that, I would argue, should focus on the molecular basis, but also in general, we need more surveillance, and we need to be able to understand both how bacteria have evolved these resistance mechanisms, understanding their ecology, and also how we use antibiotics and how that plays into our various infrastructure and clinical systems. So yeah, that's my introduction.